And our last speaker for the session is Jenny Cronin, Associate Product Manager at NanoString Technologies, who will propose solutions of spatial profiling using GeoMx fully validated antibodies.
I'm Jenny Cronin. I'm an Associate Product Manager at NanoString. I work with the GeoMx DSP system, specifically on our protein assays, and I'm here to talk about our protein offerings and our fully validated antibodies to meet your spatial profiling needs. As you've likely already heard during the conference, the GeoMx Digital Spatial Profiler uses our patented DNA barcoding technology to attach synthetic DNA oligonucleotide barcodes to antibodies or to RNA probes. And this enables you to spatially profile your tissue in any region of interest. So we can walk through our simple workflow here. On the left-hand side, you see the first step, which for a protein assay is to stain your slide with fluorescent antibodies and with our antibodies that are conjugated to our DSP barcodes. This can be done on an autostainer, or you can do it manually. Next, the system will scan your slide and you'll select regions of interest. You can select regions of interest from anywhere on your slide, and we call these ROIs. And if you want to, you can take any ROI and you can segment it, meaning you could separate it into, for example, the tumor and the tumor microenvironment. Next, in step 3, you'll see the system goes through and picks from each of those regions of interest one at a time. Or if you've segmented, it'll pick from each segment one at a time. As the barcodes are released using UV light, a microcapillary system will sip up the tags, and then in step 4, dispense them into a 96 well plate. You can then take those barcodes in our final step on the right and read them out with either our nCounter platform or on an Illumina sequencer. For our nCounter readout, we have over 200 validated antibodies. This large selection of validated protein content is bucketed into our immuno-oncology bucket with over 140 targets for human and mouse, and a neuroscience bucket with over 75 targets. For our human IO content, we are about to release 3 new panels. You'll see them on the bottom left in the IO space. Those are the MAPK signaling panel, the PI3K AKT signaling panel, and the cell death panel. Our content is designed in a core and module format, and you'll see this illustrated on the right. You have to choose a core module with about 20 plex with additional controls. And that can be a human IO core, a mouse IO core, or a human neuro core. You then have the choice to add up to 6 different 10-plex modules onto that core. And lastly, if you'd like, you can add an additional 10 custom antibodies, bringing you to a 96-plex for your experiment. Now I'd like to look in a little more detail at the IO content. For our human immuno-oncology portfolio, again, we have that core. It's here in the green table. And the core is one of our broad panels. It includes key immuno-oncology targets and markers of immune cells, including T cells, B cells, and macrophages. On the right-hand side of the slide, you'll see 4 of our 7 total modules. We'll go through the next 3 in a moment. Of these 4 modules, again, they're all approximately 10 plex. The cell typing module is the most broad. The cell typing module can allow you to profile a range of specialized cell types, including macrophages and regulatory T cells. Next, the immune activation status module can help you more deeply profile immune exhaustion. The IO drug target module includes additional checkpoint and immune modulatory targets that are currently under development for IO therapies. Some of those targets, such as PD-L1, are in the core, but this module includes additional targets that might be of interest to you. And lastly, on this slide, our pan-tumor module can allow you to more deeply profile the tumor. Here on the right-hand side, you'll see our 3 new human IO modules that'll be released shortly. They're going through our validation process now, which I'll talk about in a moment. The MAPK signaling module and the PI3K AKT signaling module will both be of interest to those who are studying targeted therapies against one of these pathways. And they both include phosphorylated protein products that measure pathway activation. Lastly, our cell death module will allow you to profile the tumor and mediators of immunogenic and programmed cell death. For our mouse models out there, our mouse immuno-oncology portfolio includes the core and 4 total modules. Those are the immune cell typing module, the immune activation status, IO drug target, and pan-tumor. Later this year, we'll also be expanding this content to match the human IO content that we've just gone over, to include those 2 new signaling pathway panels and the cell death panel. As I mentioned earlier, all of the antibodies that we offer commercially go through a thorough validation process. And I'd like to show you some data from that now. Our validation process is broken down into 5 steps shown here. On the left, you see the first step, which is an ordering screen. Here we validate our off-the-shelf antibodies in control tissue. Next, they go through a post-conjugation screen to test the specificity of the conjugated antibody. In the third step, we go through a sensitivity and specificity screen. Then an interaction screen to make sure all the antibodies will work in combination with one another. And finally, we go through a TMA screen to make sure to ensure that the antibodies perform as we'd expect in real tissue. Now, looking at each of these steps with some data from antibodies from our 3 new human IO panels. Here we show the pre and post conjugation screen. On the top row of all of these IHC images, you'll see our unconjugated antibodies, and the bottom row is the conjugated antibodies. These 4 different antibodies shown here are included in our MAPK, our cell death, or our PI3K AKT signaling panels. Our NanoString pathologist reviews all of this data to ensure that we have the same pattern of binding, both pre and post conjugation. All of the antibodies that pass that step go to a specificity screen next. During the specificity screen, we first use our nCounter system to measure the RNA expression levels in a number of different cell lines. And they're shown here in a small gray text on the x-axis. We then compare those levels to our measured protein expression levels and make sure that our proteomic profile makes sense in light of this transcriptomic profile that we've just measured. And as you can see here, especially looking between the RNA on the top graph and the protein on the bottom graph, those levels vary with one another as we'd expect. Here we show some more specificity data, again across a set of cell lines. And what I'll point out here is that in the top graph, the second and third bars there are for A431 and calliculin A cell lines. And you can see that the phospho in the yellow in the top line, the phospho MAPK ERK12 has a higher expression level in the calliculin A stem cell line than it does in the A431 cell line. Now, this is what we'd expect since the calliculin A inhibits phosphatases. Those antibodies that pass our sensitivity and specificity screen move on to an interaction screen. As I mentioned, this is to ensure that all the antibodies can work alongside one another, because again, we have this panel setup where you use a core and you add on additional modules. So here we look at our human IO core on the x-axis, and on the y-axis you see the log2 signal to noise for our human IO core plus a number of different modules. So each of the lines represented here is for a different module or a different set of modules added to the core. We do a number of different combinations, again, to ensure that the performance of the antibodies alone matches the performance of the antibodies in the cocktail of different modules. You can see the R values in the table to the right, and the correlation is high, the R values that are always greater than 0.9. Finally, all of those antibodies that have passed this test up to this point go through a TMA screen. In the TMA screen, we use tissue microarrays to look at the differential expression across different tissues, and including cancer and non-cancer tissues, and ensure that our antibodies in these core and module setups are going to work in real-world samples. So as we've discussed, our GeoMx DSP portfolio of pre-validated content in both IO and neuroscience includes over 200 different antibodies. All of these antibodies go through a robust validation pipeline so that you can use the reagents off the shelf without having to spend months or even years to validate that content. If there's an antibody of interest to your research that you've used in the past and you want to add in here, you can also do that. I mentioned you can add up to 10 different custom antibodies. You can do 10 from NanoString or 5 total from Abcam. Abcam has greater than 6,000 antibodies for custom conjugation to pick from. So you can match our protein core and our modules with that additional custom content to meet whatever your spatial profiling needs are. Our custom content goes through a different set of validation. It doesn't get fully validated as I've just laid out, but if ordered through Abcam, it does go through a pre and post conjugation screen so that you can be assured that it will work in IHC. So I'd like to leave you with that now, knowing that we have over 200 different antibodies that are fully validated, and we're happy to talk with you more about the GeoMx Digital Spatial Profiler. Thank you.
What if the antibodies I want aren't in these files?
That's a great question. If the antibodies aren't in our core or our module panels right now, then you can use custom content. So you can use up to 5 different antibodies from Abcam. And they've got over 6,000 antibodies to choose from that will work with GeoMx DSP, or you could add up to 10 custom antibodies through our NanoString protein barcoding service.
How are custom antibodies validated?
So custom antibodies do not go through the same validation process as the rest of our commercially available antibodies. If you order a custom antibody from Abcam, then it'll go through a pre and a post IHC screen. If you order a custom antibody from us, we are unable to offer the pre and post IHC screen. Instead, we ask that you only send us antibodies that have been validated by the supplier in IHC, and then we will test just like Abcam will to ensure that the oligo tag or that barcode tag has been properly conjugated to the antibody. But again, unlike Abcam, we're not able to offer a post-IHC test, post-conjugation IHC test.
Do you have any mouse neuroscience core or modules available?
We currently don't have mouse neuroscience panels available for GeoMx. However, our available human content has a decent amount of cross-reactivity, and we can provide you some information on that. Additionally, we will be releasing mouse neuroscience content at the end of this year.
What new panels are you planning?
So for new panel development, we're looking in the mouse space. So we'll be adding first mouse IO content. That content will allow the mouse IO content to match what we have available in human IO content. So that'll be adding the PI3K, AKT signaling panel, a MAPK signaling panel, and a cell death panel. Those antibodies won't be exactly the same as they are in our human panels, but we do our best to have consistency across those panels. And then we will also be adding our mouse neuroscience panels.
Will protein content be available for NGS readout?
Yes, protein content is in our plan to make for NGS readout. Currently, it's only available for encounter readout, which is why you're limited to the 96-plex currently. But we do have plans to release protein for NGS, and that will allow that plex to increase as well.
Are there any concerns that the indexing oligo interferes with the antibody binding?
That's another great question. One of the primary reasons we do our pre and post conjugation IHC is to ensure there hasn't that indexing oligo or that that barcode that we use has not interfered with how the antibody binds. So we look to ensure that we have similar patterns in those IHC stains, both pre and post conjugation.